Hi everyone, I'm very happy to have the opportunity to present our work on auto-distribution protein-protein interaction prediction for multi-species networks. Allow me to quickly introduce myself. I'm Joseph Szymborski, a PhD student at McGill University's Electrical and Computer Engineering Department and a member of MILA, the Quebec AI Institute. I performed this research with Amin Ahmad, my supervisor and professor of electrical engineering at McGill University. Professor Ahmad is affiliated with the Goodman Cancer Research Institute and MILA, the Quebec AI Institute. Be sure to reach out online through Mastodon or my personal website. Let's begin with a brief introduction to the problem we're trying to solve. We're primarily concerned with protein-protein interactions, or PPIs. Most of what cells do is, in fact, achieved by proteins making physical interactions with one another. When proteins interact, they can chemically or physically modify each other. And to the great delight of the computer scientists in the room, they can be thought of as an undirected graph, where each protein forms a node and their interaction is the edge between them. It's an incomplete model, but it's very helpful and has gotten us pretty far historically. So this brings us to the question, how do we determine whether two proteins interact? Traditionally, this is done using so-called wet lab experiments, like co-immunoprecipitation, which is done at the lab bench. These experiments can be very slow and very expensive, but they do constitute hard evidence of a protein-protein interaction. There is, however, a major consequence of the slow, expensive nature of these experiments, namely that it's prohibitively expensive to exhaustively evaluate all protein pairs across all species. This means that while well-studied model organisms have a rich wealth of data to mine, there's a great many less studied models for which we have very little to no PPI data. In fact, we've only conducted protein-protein interaction experiments on a very small proportion of species. String is a comprehensive database of protein-protein interactions for which we have evidence. Only 30% of taxa at the class rank and just over half as many at the order rank are present in the string core database. Clearly, there is still many species for which there simply isn't enough PPI data. Another way to predict protein interactions is using computational models. Relative to bench experiments, computational models are much faster and far cheaper. They are, however, simply predictions and not evidence of protein interactions. The speed and cost of inferring protein interactions using computational models make them a particularly good candidate for addressing that species gap that I just described. While computational models seem to be a great answer towards addressing the species gap, there is a bit of a complication. Firstly, good ML models require lots of high-quality data to make accurate predictions. Annoyingly, these neglected species that would best benefit from ML models that predict PPIs don't have enough or often any data to train on. To get around this predicament, we'd ideally want a model capable of cross-species predictions, where the model is trained on a species for which we have a lot of data and is still capable of making predictions on a different species for which we do not have sufficient data. The ability for a model to generalize to auto-distribution predictions is difficult for many models. The challenge in making generalized PPI prediction models has been known for quite some time now, and was illustrated quite well by Park and Marcotte as early as 2012. When we construct datasets for training and evaluating machine learning models, one may take a naive approach and randomly assign interactions to a training or testing split randomly. Park and Marcotte found that with such an evaluation scheme, methods performed quite well across the board. However, if we were to call a set of proteins training proteins and another set testing proteins, and then removed any interaction between proteins that weren't of the same type, such that no training proteins are found in the test set and no test proteins are found in the training set, we find that the performance of those same methods degrades significantly, often dropping to near random. And while the Park and Marcotte paper was from 2012, these findings have only gotten more pertinent with the rise of deep learning methods which have higher tendency of overfitting. This 2023 preprint by Judith Burnett et al. Attributes the near perfect performance of many such PPI models to the data leakage caused by random splitting. So we've established there's a species gap, and that to cross it we need a model which generalizes to the cross-species PPI prediction task. We've also established that making generalized models is hard. Well, let me propose our method, RAPPPID, as a possible solution. RAPPPID is a deep, regularized twin neural network that adopts a modified AWD LSTM. RAPPPID considers pairs of amino acid sequences with an interaction label, making RAPPPID a sequence-based method. The amino acid sequence of both proteins are first tokenized with the sentence piece tokenizer. We then compute fixed-length vectors that represent each sequence using a bidirectional AWD LSTM. Latent vectors are then concatenated and are inputted into a two-layer fully connected classification head. The output of the classifier is the interaction probability. To learn more about the RAPPPID model, please see our paper in bioinformatics. RAPPPID is hardly the only twin network for PPI prediction, but there are some notable techniques we've adopted to help regularize the network. We've adopted a regularized LSTM called AWD LSTM. We use embedding dropout, the Ranger 21 optimizer, and stochastic weight averaging, SWA. Last but certainly not least, we've adopted the sentence piece tokenizer. I do, however, want to spend some more time on how the datasets we created for RAPPPID were made. Going into this project, I thought I'd be spending a lot more time on tweaking model architectures, but ultimately, making sure our datasets were strict and our evaluations fair is what took most of my time, and was an indispensable part in making RAPPPID generalize. In creating strict datasets for PPI prediction, we first made sure to split both proteins and their edges in the way I described earlier in the talk. Another important aspect was ensuring that proteins are no more than 90% similar between training and testing splits. This gives us a guarantee that the amino acid sequences of any protein in the training set differs by at least 10% from any amino acid sequence in the testing set. To achieve this, we take advantage of the UniRef database, which precomputes the similarities of proteins and clusters them by percent similarity. Finally, because we are using a supervised algorithm, we need both positive and negative samples. Negative examples are difficult to come by when it comes to PPI data, and it has been shown that most methods end up resulting in a biased distribution that is easy to overfit to. Uniformly sampling negative pairs of proteins not known to interact randomly has shown to be the best method to obtain these samples. We had to create a lot of these datasets, and we needed a neat way to do this in a repeatable and reliable way. That's why we developed a convenient tool that we will be sharing to create these datasets from scratch. You can download and process third-party datasets like the string and UniRef 90 datasets for humans. You can make datasets for training RAPPPID with all the characteristics we discussed. And you can convert those RAPPPID datasets into datasets suitable for use with other PPI models like Descript and Sprint. So without further ado, I'm happy to share some of our results. We find that RAPPPID performs better than leading PPI prediction methods when making predictions on a dataset of human interactions. This dataset was prepared using the protein splitting approach outlined in the methodology section. Here we see that RAPPPID obtains higher scores than leading PPI prediction methods, obtaining an average AUROC of 0.767, average precision of 0.779, and a Matthews correlation coefficient of 0.371. These averages were obtained from evaluating these methods on 3 random training and testing splits. We were also happy to learn that RAPPPID learns biologically relevant features. We tested whether RAPPPID was actually relying on the sequences of the proteins in the PPI graph, or rather taking advantage of characteristics of the PPI graph like degree distribution. To do this, we randomly assign proteins the amino acid sequences of different random proteins. This maintains the degree distribution, but the network no longer makes any biological sense. A model that relies on the biology encoded by the protein sequences should perform well on the string dataset, but perform poorly on the scrambled string dataset. RAPPPID's performance as measured by AUROC drops by 43%, down to random chance, when trained and tested on the scrambled dataset, indicating that RAPPPID relies on biologically relevant features. We also find that RAPPPID maintains high intraspecies performance on various organisms. We trained and tested RAPPPID on 8 distinct organisms, ranging from mouse to Drosophila to baker's yeast. Here we find that RAPPPID achieves AUROC and AP scores that are largely comparable across the board. A result we were quite pleased to see was RAPPID's cross-species performance. This graph illustrates that a RAPPPID model, which is trained on model PPIs, maintain its performance when tested on other evolutionarily distinct species. On the y-axis, we have Matthew's correlation coefficient, and on the x-axis we have the evolutionary or patristic distance relative to Homo sapiens. The unfilled markers here represent the performance RAPPID achieves when trained and tested on the species indicated by the marker color. The filled markers represent the performance of RAPPID when trained on human data and tested on the species indicated by the marker color. The dashed gray line indicates the performance of the human model when tested on human data. As you can see, the cross-species performance appears strong across nearly every organism, independent of evolutionary distance. We're also very excited to be taking RAPPID to the internet. We're in the process of bringing online a RAPPID inference portal where you can input your own amino acid sequences and get back and return interaction probabilities. In our preliminary tests, inferring the interaction between a user-submitted protein sequence and all the human UniProt KB protein sequences takes about a minute to complete. In conclusion, we established that PPI data suffers from a species gap, that we need models capable of cross-species predictions to bridge that species gap, and that in order to do that, we need models that generalize so we can make accurate cross-species predictions. RAPPID is one such generalized model, and it outperforms leading PPI prediction methods. RAPPID also makes accurate cross-species predictions. We established that RAPPID's predictions are indeed based on the biology encoded in the amino acid sequences and not something like network topology. And that RAPPID is computationally efficient, and preliminary results show that it is suitable to run an internet service. Thanks to the members of the Combine Lab for their feedback and support, and thanks to our supporters. Thank you so much for listening, and have a great conference.